Hi, I'm Tiffany Luong, a PhD student studying bacteriophages in the Roach Lab at SDSU. And now that we've gotten to know each other a little bit, let's take a look deep within ourselves, not emotionally, but rather microscopically. So our body is a home to an amazing variety of microorganisms. In fact, these microorganisms outnumber our cells by 10 to 1 and comprise of what we call the microbiome. Now, to introduce you to some of your friends in the microbiome, we have both commensal and pathogenic bacteria. Now, commensal bacteria are beneficial bacteria, which do helpful things like help you convert food into energy. And they also do a neat thing of occupying areas where pathogenic bacteria would otherwise use to infect and cause disease. And although this may sound scary, we're actually also home to a variety of viruses, which we call the virome. The virome consists of both eukaryotic viruses, such as herpes, which, when it acutely infects us, uses your cell to create more copies of itself. And usually when this happens, we also experience some type of disease. But what our bodies have evolved to do is we've co-evolved with some of these viral communities to reach a state of equilibrium, where the viruses are still maintained at up to 10 to the 12 in our body, but are no longer causing disease. In addition to eukaryotic viruses, what I'm more interested in are prokaryotic viruses called phages. We have upwards of 1 billion phages in our bodies at all times. And these phages are super interesting because they are not capable of infecting human cells, but instead infect and use bacteria to create new viruses, thereby helping us eliminate a lot of pathogenic bacteria that can reside within the body. But my area of interest is in the crosstalk between phages and with our cells. Since phages can't infect our cells, what exactly is the crosstalk here and how are they interacting? Human cells are able to sense all these different microorganisms through receptors that we call toll-like receptors. TLRs can recognize the different DNA, RNA, or molecular patterns from these microorganisms and translate it into an immune response. This tells your body whether it needs to protect itself against these microorganisms. But what we don't know right now is what role do phages play? If and when TLRs sense phages, what sort of response is elicited? I'm really interested in this topic because phages have been implicated in a variety of diseases, such as inflammatory bowel disease, where there's a significant difference in the phage populations between healthy and IBD patients. Does this mean that the response between phages and human cells is one that leads to homeostasis or one that leads to disease? Hopefully, finding the answer to these questions and more can help us develop novel therapeutics to tackle IBD and diseases like it, as well as help us understand our tiny but helpful bacterial virus friends a little better. Thank you.